Okay. Yeah, I apologize. I have a bad throat, so I might end up coughing through most of my talk. All right. So I think this is a really interesting topic. It's something that it's only been in our in kind of the on the radar of people doing statistical genetics over the last 5 to 10 years, and I want to give you a kind of a flavor of the problems and kind of the challenges involved in at the intersection of doing private genomic analyses. By the way, can you hear me at the back? No? Okay. All right. So let's start with a motivating example. So this is GWAS. You're looking for association between traits and SNPs, and typically most GWASs look at common SNPs, which means they have frequencies above some threshold like 1 or 5%. And most of these SNPs from our current catalogs of GWAS associated variants have very low effect sizes. And so they explain a very small proportion of the variation in a given phenotype. And so the consequence of that is that we need to be testing of the order of millions of SNPs to find effects. And we need to be doing this over large collections of samples to be able to have the power to actually make these, to discover these associations. Right? So kind of you have to play this game where you're looking at larger and larger data sets to be able to find statistically significant associations. So the way this has been handled in the GWAS community is by combining analyses across different data sets that have been collected by diverse groups. And this is what people typically call meta-analyses. And so here is a typical example of this. So here is a meta-analysis that was published about 5 years ago, where people looked at height. As your height is your classic polygenic trait. So it has been shown to have lots of variants, each of which has a very small effect associated with height. And so in this example study, they looked at close to 200,000 individuals, and they did this by pooling data from multiple different data sets. So clearly there's this kind of realization that if you want to get at the genetic basis of complex phenotypes, you need to be sharing data across different data sets that have been collected. But the catch with trying to be able to do this is that privacy becomes a major concern. So there's a question of how do you actually share data while still keeping the privacy of individuals who have contributed to your data set secure. So here is an example from a study which tried to survey what are people's attitudes about sharing of data versus privacy. And so basically they polled a bunch of people and asked them how willing are you to actually contribute your DNA for research, and how much do you care about your privacy being protected. And kind of the key thing here is a vast majority of people strongly agree that they both want to share research and they want to protect privacy, right? And that brings with it the inherent conflict. And one thing they asked is, okay, now let's say you had to choose one or the other. You had to choose either between privacy or between data sharing. Then suddenly there's a little bit of a flip where people say, oh, okay, I'm willing to share my data, even at the expense of some privacy. And they ask some further questions, and they realize that the reason that they are able to do this is because they kind of think that, these kinds of privacy threats are not immediate, they are way down the future, so they won't personally affect me. So clearly there's this tension going on where people want both, and the question is it even realizable? Is it feasible to meet both ends? And what are the kinds of threats that are realizable? Okay. So my talk has kind of two basic parts. The first part is I'm going to be looking at techniques that people have realized that get around privacy protections in data sets. And the second part is some techniques which mitigate against that. And I'd like to kind of work towards one specific model, which I think of as a coherent model for privacy, which is called differential privacy. And again, I just want to kind of end. I mean, the bottom line of all this is still very much open research. Differential privacy is a very interesting and solid foundation to build on, but it's unclear that it actually gives you the right kind of privacy utility trade-offs. Okay, so there are a couple of different ways of thinking about the different kinds of attacks that can breach privacy. And so this was outlined in a very nice review paper by Yaniv Erlich and Arvind Narayanan. And so there are a couple of entities that we're dealing with in the case of genomic data. So the first thing that we're dealing with is a person's DNA. The second entity we're dealing with is identity, so name, other identifiers like their zip code, date of birth, and so on. And the third entity we're dealing with is phenotype, right? So we like to predict whether or not you have disease. So so the first form of attack that people have considered is, let's say you have the de-identified DNA of an individual. Most data sets have de-identified DNA. Can we go from de-identified DNA to getting at identity, right? So can we tie these genotypes or genome sequences to individuals' names and other information? And so this class of attacks is what's going to be called identity tracing, and we'll talk a little bit about what flavor these identity tracing attacks come in. The second is, once you've identified a person's DNA, now you like to go and say, can you say what the person's phenotype is? Does the person have a certain disease? And these go by the name of attribute disclosure, because the phenotype is an attribute of this individual, and so you'd like to breach or disclose that specific attribute. And so we'll talk a little bit about attribute disclosure as well, and spend a fair bit of time here, because I think this is where a lot of interesting open problems lie. And finally, there's something called completion attacks. I won't spend too much time on this, just briefly give you some ideas of what these completion attacks enable. Okay, so let's start with identity tracing. So as I said, the idea is you want to go from a person's de-identified DNA to some notion of identity. And so the key things that you do here is you look for what are called quasi-identifiers. So quasi-identifiers are basically attributes which in themselves have very little power to identify, but in combination can identify individuals. And basically, you have these quasi-identifiers often available as metadata in genomic studies. So often you have covariates like age or sex, which you want to use for correcting, and these often serve as quasi-identifiers. So here's an example. Let's say you look at these three quasi-identifiers: date of birth, sex, and zip code. Each of these individually doesn't have a lot of resolution in telling who the individual is, but together it's been found, looking at, say, census records, that about 60% of US individuals can be uniquely identified by date of birth, sex, and zip code. So that tuple, this three tuple, is what we'd call a quasi-identifier. So here's kind of a classical example of how you have individual attributes or pieces of information which don't identify, but when you connect them up, you can actually get identifying information. So this was a study that was published in 2002, and what's happening here is, so this was an attack that was based on data from Massachusetts. So they were looking at medical data. So there was this insurance company in Massachusetts which published a whole bunch of identifiers, so all of these identifiers, under the assumption that these are not identifying. So as a researcher, you could go to this insurance company and buy a data set which had all these identifiers. And what this researcher did was took these identifiers and intersected them with publicly available voters list in Massachusetts. Okay, and so the voters list have all these other identifiers, and so in common they have zip code, birth date, and sex. And what they found was they could identify the Massachusetts state governor William Wells' medical record by intersecting it with the voters list, right? So it turns out that there are so they knew the zip code where the governor lived, and so you start with about 54,000 residents, and 6 of them share the governor's birth date. Of those, 3 are men, and 1 had the zip code, right? And using that, they could actually pull out the governor's health record. So this was an example where, by using these quasi-identifiers, each of which is kind of weakly identifying, you can actually gain power to reveal identifying information. So this kind of attacks have only become more problematic as you have genetic data sets. And so one way to do this is a methodology that's called genealogical triangulation. So the basic idea is you now have data sets, large genealogical data sets, because people are interested in genealogies and where they came from, which also have genetic information and other kinds of identifying information. So you can tie both genealogical and genetic information to start identifying individuals. So one very clear example of this is using Y chromosome genealogies. And the basic idea behind Y chromosome triangulation is, so Y chromosomes are paternally transmitted markers, so they come from your dad's dad. And the other thing that also gets transmitted in a paternal lineage in most societies are your surnames, right? So by trying to match up Y chromosomes of individuals with Y chromosomes of people in genealogies, you can actually figure out a person's surname. That's the basic idea. So here is an example. You have a pedigree. So this is a genealogy. And so these are the pedigrees of the Smiths, and these are the pedigrees of the Johnsons. And so you see that they get transmitted, these surnames get transmitted in the paternal lineage. And now you have the Y chromosome of the target you're trying to de-identify. And so you take this target's Y chromosome and you essentially match it up against the Y chromosomes of people in these genealogical databases. And you're essentially asking whether you match the Smith pedigree versus the Johnson pedigree. So the reason these kinds of attacks are now feasible is you have large data sets of genealogical information based on the Y chromosome. So each of these have of the order of hundreds of thousands of records. So for example, one such data set is called the Y-search data set, but there are many of them. And so if you combine over all of them, I think right now we have more than 500,000 records aggregated across these genealogical data sets. And they allow you to query. So all you need to do is you need to have your target's Y chromosome information. Often these are short tandem repeats that people are interested in. So once you have a person's STR information, you go and enter it into a web interface, and it'll tell you what the matches are. So it's really, really simple. So essentially, the data you're dealing with is these STRs, so not SNPs. And so these STRs are basically places in the Y chromosome where there are variable number of repeats across people. Right? So there'll be some people who have 4 repeats at this STR, another person might have 6 repeats, and you're just looking for matches in the number of repeats at a number of these STR loci. And so the statistical question you're trying to answer is you're trying to find the match with the closest time to the most recent common ancestor for your target haplotype. Right? So remember, the Y chromosome is non-recombining, so it's just one single locus. And so you have one single TMRCA that connects your haplotype to a potential match. So you have your query and you have a match, and there's a time that you're trying to estimate. And so essentially you're looking at some number of loci, n loci, typically anywhere from 30 to 80 STRs. And the data you're looking at is a match at each of these. So at each of these n loci, you're asking whether you have a match or not. And so that's your data. And given the time to the most recent common ancestor, given t, and the mutation rate, so given t and mu, for a given locus, you can ask what's the probability of a match. So that just turns out to be what's the probability that there were no mutations during the time to the most recent common ancestor. Right? So it's a Poisson process of mutations on this genealogy. And so the probability that there are no mutations is simply given by 2 times t times mu being the rate, and this is the Poisson probability of there being no mutations. All right? So now you're actually dealing with many of these STR loci. So if you observe matches and mismatches at many of these STR loci, you can then compute what's the posterior probability, if you're Bayesian, of the time to the most recent common ancestor, given the match statistics, given mutation rates, and some estimate of the effective population sizes on the Y chromosome. So these two are are often known from previous data sets. This is the data you're observing, and you can compute the posterior probability. Right? So very simple statistics that you can do to actually back out these quantities. So, so this was done. This was first demonstrated on an individual level in this paper from Melissa Gimrek et al. And just to give you a little bit of idea, what they did is they looked at these TMRCAs, and you can put thresholds on when you declare whether an individual is in a data set or not. And as you vary the threshold, you're going from a very lenient threshold to a very stringent threshold. You're looking at different probabilities of getting a true match, and you're trying to control the probabilities of getting a false match, right? And you can figure out a reasonable place to balance these two, and they chose some threshold here. So so for example, one statistic they came up with, they ran some simulations. They showed that about 12% of Caucasian males are at risk for these kinds of Y chromosomal triangulation attacks based on the data sets they consider. And this was a couple of years back, but you can imagine as these data sets get more and more individuals, that number goes up. And at those thresholds, they have in 83% of the case, 83% of the time you make a query, you get a result which says it's ambiguous, and you have a 5% false positive rate, and the rest is your probability of actually having a successful hit. So the surnames that they do identify, they find that they are shared by less than 400,000 individuals. And to give you a kind of a rough estimate, this is as informative as getting the zip code of an individual. So that's about the same level of detail that you have. So again, as I said, each of these pieces of information, like your Y chromosome, it may not be individually identifying, but the question is, what can you do when you have multiple pieces of information? And so here is one way of seeing this. So you're starting with an unknown genome on the left, and you're trying to get to the identity of the individual. And you can see this by asking how many bits of information is contributed by each of these pieces that you have, right? So the bits of information is simply minus log of the probability that this information is present. So for example, there are about 300 million individuals in the US. So you're starting out with a space that's about 28 bits long. And if you know the sex, that's 1 bit, so that reduces it to 27 bits. If you know the state of residence, that reduces it by another 5 bits. Age, again, another 6 bits. And then if you know the surname, that reduces it by another 13 bits. Right? And then if you have further kinds of information, you can actually go all the way. So that's telling you how a person would actually be able to launch one of these triangulation attacks. So they actually, so one of the nice things about this paper, and one of the things that made it so interesting, was they actually did it on individuals who were publicly identified and had the genome sequenced. So they looked at Mike Snyder's genome, they looked at James West, and they looked at Craig Venter. And essentially they ran these individuals' genomes through their pipelines to see if they could be identified. So what you're seeing here on the x-axis is the matches that they get back, and along with the TMRCAs. Right? So in the case of Snyder and West, you see that there is no unique match that stands out. They have a whole bunch of matches that are about the same in terms of the TMRCA, and the TMRCA is about 20 generations. On the other hand, Venter is different. There is one clear match at about 8 generations of TMRCA, and then you have a whole bunch of other surnames. So and of course that the nice thing is that is of course the surname associated with Venter. So again, so this was saying that for a small but not insignificant proportion of your population, these kinds of attacks are actually feasible. All right. So an open question in this field is this is all with Y chromosomal data, but surely we have autosomal data now. That must be giving us a lot more power, and people are of course depositing autosomal data into these genealogical data sets. So that's an open question. We don't know how much additional power can be gained by using autosomal information. All right, so now let's go to the second attack. So let's say you have an individual and you've identified them. You'd like to learn about some potentially bad phenotype. So the setting is you have the genotype of an individual, and let's say you have a data set of genotype attribute information, genotype phenotype information. You can just go ahead and scan this and get a match, right? And of course, if you have GWAS data, if you have genomic data, this is trivial. People have estimated that you just need about 300 random SNPs, and that's enough to get a unique match in the population. So definitely with 500,000 or any reasonable GWAS, this is easy identification. So this is why the community as a whole has put a lot of individual level genotype data behind access control walls. So you have this, if you go to a place like dbGaP, you have this two-tier access system. There is restricted access to individual level data, but at least till some time back, and now it's again opening up again, we have public access for summary statistics. So if you do a study, the p-values or the effect size estimates are often made publicly available. The individual level data goes behind dbGaP. So the question is, how secure is this? Till about 2008, people thought that this was a fairly good way of handling the privacy utility trade-off. And that of course changed when there was a paper that came out of David Craig's group that showed that even with this public summary statistic data, you could actually identify individuals in data sets. So here is how such an attack would look like. So you have a case control data set, and what you have are summary statistics, which are the allele frequencies of a SNP in cases and controls. So this is the public data, and this is private. And the question we are asking is, if I have a genotype, is this in the case group or in the control group? So it's a very simple question. And the implication is, if I can say in the case group, then I know that what phenotype you have. You have the phenotype that's shared amongst all cases. So this paper from David Craig's group was the first to show that these kinds of attacks are actually feasible with the kinds of data sets that are available today. So the way I want to show you this is just to show you one figure from the Homer et al. Data set. And it's actually, a priori, it is something that's hard to believe. It's not intuitive. In fact, most of us, when this paper came out, thought that they made a mistake. But it turns out it's actually true that you can actually identify individuals in these kinds of data sets. So okay, what are we seeing here? On the x-axis is S, which is the number of SNPs that are made publicly available. And think of F, or think of 1 over F as your sample size, right? So you're going to bigger sample sizes here, and you're going to more SNPs here. And the color here is negative log 10 of the p-value with which you're able to identify an individual. All right, so green means you're more confidently able to identify an individual in this data set. So looking at this, if I have 10,000 SNPs and a data set of size 1,000, I can identify somebody with about 10 to the negative 3. But if I now have 100,000 SNPs, that becomes 10 to the negative 8. So that's almost certain identification. So of course, as soon as this paper came out, there was a big reaction, and a lot of public data repositories, the NIH, the Wellcome Trust, removed their summary data. And so we went from this world where there was very little access to summary, there was a little bit of access to summary data to very little data. And that led to a lot of debate about how this is bad for research in general. And one of the things that, again, this debate brought to the fore is when we actually talk about privacy and what's possible to do privately, we don't do it in the context of a mathematical model, right? So we assume that these ad hoc measures give you some sense of privacy, but what you actually want is a clear model which says these are the assumptions that you're making, and this is what is feasible. So I'm going to tell you a little bit about some work that we did on this problem. This was a while back, and trying to quantify what are the privacy guarantees that you can give in the context of these individual detection attacks. So here we're now going to be classical frequentists, and so we're going to formulate individual detection as a hypothesis testing problem, right? So so it's basically going to be a likelihood ratio test, where the two hypotheses you're looking at is this individual in my data set or not, right? And so H0 is the individual is not in my data set, H1 is the individual is. And X is the individual that you have. So you're testing these two hypotheses, and this is the likelihood ratio test, right? And again, classical statistics 101 tells you that the optimal test for a test that compares two simple hypotheses is given by the Neyman-Pearson lemma, right? So you look at the likelihood ratio test as your test statistic. You ask if it's bigger than some threshold, which you set based on your false positive rate alpha, right? And that is the test that gives you the optimal power, right? So what's the idea? The idea is this is the best test that you can perform under this situation. So if I were to be able to quantify the power of the likelihood ratio test, it's an optimal test, so that gives me an upper bound on the power of any other method to be able to do individual detection, right? So that can tell us or give us some guidelines for how many SNPs can be exposed if you're willing to put a bound on the power and the false positive rate. Okay, so here's how we're going to formulate it. So that's the null, that's the alternate. So you have you're looking at a single SNP here. So P is the allele frequency of at that SNP, and in the null we're drawing n individuals independently at random. These xi's are basically Bernoulli draws from this allele frequency, and of course this is private data, so you don't get to see that. What you instead get to see is the summary, so that's P hat, right? So this is the summary statistic of the allele frequencies at that SNP. And because it's the null, you have another individual who is not part of this data set, right? So this is your target individual, and you're trying to identify them. The alternate just has a slight difference. In the alternate, you have the individual you're trying to identify being part of the pool, right? So very small difference, and that's all there is. And now in practice, you have m possible SNPs. This is just one SNP. You can have m of these, and you get to see the same process across them. So now you can write down the likelihood ratio statistic. So this is what the likelihood ratio statistic looks like for m SNPs, and this is what you see for each SNP, right? So this is basically what you're seeing in the alternate versus what you're seeing in the null. And this is just basically you can work this out by doing Bernoulli sampling. So it's very simple. All right. So it seems like we have solved the problem, right? We have written down the likelihood ratio statistic, and all we need to do now is figure out the power. It turns out that that's not the case, because the power actually depends on this quantity P sub j, which is the allele frequency of this SNP in the population. But we don't actually have P sub j, we only have estimates of P sub j, right? We don't know what the true P sub j's are, right? So it doesn't give us an optimal, it doesn't give us a bound on the power of the optimal method. So so here is where there is some technical information that we can use, and the basic idea is we looked at what happens when you have pools that are large, when your data set size or the number of individuals becomes large. And when that happens, basically you can do asymptotics, and I won't get into the details there, but it turns out that you can write down what is the expectation and the variance of this likelihood ratio statistic under the null and the alternative. And in this asymptotic regime, what happens is the expectation and the variance don't depend on the allele frequencies, right? So it only depends on n, which is the number of individuals in your pool, and it's going to depend on m, which is the number of SNPs. So it turns out, to a first order approximation, all you care about is m and n, which are your kind of design parameters. Okay. So a couple of assumptions going here. Firstly, the asymptotics needs to be valid. The second is, again, for the asymptotics to be valid, the SNPs need to be common. So if you have SNPs that are singletons, for example, these asymptotics wouldn't hold. And finally, you need SNPs to be independent. So you need to be, you're looking at independent draws from this process. All right. So under these kinds of assumptions, of course, we can figure out what is the optimal power for any of these methods. And it turns out it's a very simple expression. So here is what the distribution of the likelihood ratio statistic looks like under the null and under the alternate. So asymptotic, so everything is Gaussian. And so the null has some mean and a variance, and the alternate has a mean and a variance. And your optimal test is going to say, this is a place where I'm going to put a threshold, and if the likelihood ratio test is to the right of that threshold, I reject the null. Right? So for a given threshold, you can ask what's the false positive rate alpha, and what's the false negative rate 1 minus beta. So all of these are just fallout for us. So if I were to give you an alpha and a beta, we can basically write how the quantiles of alphas and betas relate to the two parameters we care about, m and n. And it's basically this simple expression. Right? So any questions? So what this gives us is, you pick your alpha, you pick your beta, which is acceptable, and this gives you the m and n that allows you to achieve those alphas and betas. So here are some numbers that you can plug in. So you start with alpha like 10 to the negative 2, negative 3, 10 to the negative 4. Beta is say 10 times that. And so for these values, you get different values of m over n. Right? So it gives you some hope for actually figuring out what is privately releasable. Okay. So we can do additional fancier stuff. We can do things like what happens when you want to apply this likelihood ratio test in practice. So let's say you don't believe the assumptions that we're making, but you'd actually like to test it out in practice on a real data set. And in those kinds of settings, you can do things like a leave one out procedure to estimate your power. So the idea is you look at each individual, pull them out of your data set, and consider a data set which has the individual versus one that does not. And with that, you can now you can use estimates of the population allele frequencies from an external data set. Right? So under that, you can actually get an empirical likelihood ratio test. So this is more technical. So I'm just going to go look at some simulations. So we just did some simulations here, where you're looking at the false positive rate on the x-axis, the power on the y-axis. And so we looked at 1,000 SNPs and 10,000 SNPs in simulations. And we're looking at, say, the likelihood ratio test in theory. So this is the dots are the likelihood ratio test in theory. This is the likelihood ratio test in practice, and this is the test that was proposed in the Homer et al. Paper. And so what we see here is roughly all of these are giving you the appropriate kind of behavior. And what we can do is now we can say, okay, for a false positive rate of 10 to the negative 3, what is the appropriate power, and is that a reasonable release mechanism? You can also consider all kinds of extensions of this. What happens when you have dependent SNPs? What happens when you have genotyping errors? What about relatives, and so on? And we can actually extend the theory into those settings. People have looked at other kinds of extensions as well, going beyond just case control GWASs. So this is a paper from Nancy Cox's group, where they looked at doing these kinds of attacks when you have quantitative trait GWASs. So you don't have allele frequencies, you have actually effect size estimates. And kind of the bottom line is everything looks about the same. So you can write down the likelihood ratio test. You have a similar expression that depends on m and n, but now it has some extra stuff that depends on the phenotypic variance. And it turns out that you can do the same kinds of tricks in the context of quantitative traits. Another attack that was considered, this was from Eric Schatz's group, was you have gene expression data. So you have publicly available gene expression data from a whole bunch of different sources, and you've also identified EQTLs. So can you use these EQTLs to predict genotypes from gene expression data and use that to perform identification in different data sets? And so here you can't see the colors coming out too well. But what they did is they took one gene expression data set on the left. So this was this was in liver that they looked. And they used that as their training data set where they identified these EQTLs. And then they used those EQTLs and the genotypes to actually perform identification in a test data set. Right? And this is hard to discern from here, but at all places which don't have these whites, those are places where they're actually exactly able to match up individuals across the two data sets. So what they're showing is for pretty much most of the individuals in these data sets, they can match up who are the individuals based on their genotype data that is inferred from gene expression. Right? So that gives us another way of actually launching these kinds of disclosure attacks. All right. So I just want to say one word about completion attacks because they're interesting, but they've already been covered. So the completion attack is you have genotypes from some part of an individual's genome, or maybe not from the individual, but their relatives. Can you actually infer the individual's genome itself? And of course, we had talks on imputation, and that's precisely what genotype imputation is doing. Right? So that's exactly what completion or genotype imputation allows us to do. And as a kind of an adversarial way of using imputation was this paper from Peter Vischer's group, which showed that so when James Watson had his genome sequenced, he didn't want to know about the status of the ApoE gene, which is associated with Alzheimer's risk. And of course, you can use imputation to fill in the status of the ApoE gene, and that's exactly what they did in this data set, in this paper. Right. So I guess that's kind of covering the range of these attacks, and I guess the kind of the punchline here is trying to reason about what you can do given all these different data sets is extremely complicated and messy. So let's now spend a few minutes looking at what are the possible ways of mitigating some of these attacks. So I talked about one, which is just restricting how much data you present. Right? If you present no data, of course, you're perfectly safe. But there are ways of actually quantifying how much you lose by releasing subsets of these data. So that's one way of going about it. So that's, I guess, access control of different kinds. There are a few other techniques that I want to just briefly mention. So one is called an anonymization. So this is basically doing summarization, but at a smaller granularity. Instead of summarizing over your entire data set, you summarize over subsets of individuals so that you cannot tell whether one of those people in the subset is actually identified. So one way to do it is called K anonymity. So there's a parameter K. And the idea is you aggregate the records in your data set so that there are at least K minus 1 other people who have the same set of identifiers. So you cannot differentiate amongst any more than one of these K people. Right? So here is like a table, and you have quasi identifiers, which are these 4 columns: race, birth, gender, and your zip code. And this table has been too anonymized. So which means if you look at these 4 columns, they're always found in at least 2 people. Right? So even if you manage to identify an individual, you can only say this is one of these 2 individuals. So that has worked okay in the database setting, but it works much less well in the GWAS. And people have found that this is not a very sound way of actually giving privacy guarantees. So instead, a technique which has a lot more of a theoretically sound framework is called differential privacy. Right? So last few minutes, I just want to spend talking about differential privacy. I think it's a very interesting, really hot area of research, but I think there's still lots to be done here. So the basic idea of differential privacy is instead of restricting how much information you release, you add noise to it. And you try to calibrate the amount of noise you want to add so that it gives you some level of privacy. So here is the basic framework. you're starting with your data. So you have, say, x1 to xm, that's your individual level data, and you're computing some function which is going to be your private function. So in the case of GWAS, think of f as the summary statistics that you want to release at every step. Right? So the basic idea, as I said, is you don't release f, which was problematic. You add some noise to f, and that's what you release. So you release f tilde. Okay. So again, it must still be useful for everybody else but the attacker. So the question is, what is this epsilon, or what is this noise that you're adding? So this f tilde is said to be differentially private. So not every f tilde is differentially private if it has a certain property. Right? So what's this property? So you take two data sets, so x and y. Okay, so these are two GWAS data sets, and they differ in a single individual. So delta xy is 1, which means there's just one individual who differs between the two of them. And you apply your f twiddle to x, and you apply to y. Right? So you get two different f twiddles, and you ask how different are the distributions of these two different f twiddles. So that's the quantity on top. Right? So what's the probability that it belongs in some set? And the differential, it's said to be differentially private with a parameter that's given by alpha if these two distributions are not much more different than this x of alpha. Right? So the way to interpret this is think of alpha as how differentially private you want to be. The lower the alpha, the more differentially private. Right? So let's say I set alpha to be sorry. So let's say I set alpha to be 0. Okay, so that means I think I have a mistake somewhere here. Oh, that's fine. Yeah. So if I set alpha to be 0, right, that means the probabilities of these two quantities should be less than or equal to 1. So, and it actually turns out that it lies within a window around the alphas. So that's something that you can show here. So what that's saying is for small values of alpha, that these probabilities are actually going to be close to each other. Right? So you pick a value of alpha, and if f twiddle satisfies this relationship, then it's said to be differentially private with a parameter that's given by alpha. So putting it back into this likelihood ratio test framework that we started out with, if I were to design a test for testing if an individual is in a data set, and this test has a false positive rate given by this, then if it is differentially private, the power is at most gamma times e of alpha. So if I set alpha to be really, really small, then the power also becomes very small. And so you have a differentially private mechanism. So that's an abstract definition. How do you actually get such a mechanism? So one way is something called an exponential mechanism. So the idea is simple. For every function that you're releasing, you compute something called its sensitivity. So the sensitivity is basically telling you how much the function changes if you change one entry in its input. So that's S of f. And you add noise, which is Laplace, with parameters that depend on alpha, which is your differentially private parameter, and the sensitivity. Right? So the more sensitive your function is, right, this is Laplace, which means you're going to have a bigger standard deviation around your error. Similarly, the smaller alpha is, the more differentially private is, you're going to add a bigger standard error. So you're adding more noise if you want more privacy, or if your function is more sensitive. So this is what sensitivity is formally. So sensitivity of this function is you take the function and evaluate it on two different x's and y's, two different data points, and you compute its L1 norm. And the question is, what's the maximum possible L1 norm across two x and y's that are separated by 1, so that are neighbors in this sense. Okay, so this is again abstract. What happens when you apply to GWAS? So in GWAS, the f is the allele frequency computed across all the SNPs in your GWAS. So we need to basically figure out what is the sensitivity of the F. So what would the sensitivity be? Ballpark. So think of a data set where you take one individual and replace it with another who differs from the other individual at every SNP. So you have an individual with all zeros and another individual with all ones, right? So the allele the Fs that you're computing is going to differ at every SNP, and so the sensitivity is going to be order the number of SNPs. So that's the bad news. The bad news is you're essentially adding noise which is proportional to the number of SNPs for you to get differential privacy to work. So it's a great concept, but it doesn't work. It basically washes out all the signal in your data set. Yeah, sorry.
Why choose the L1 norm?
Yeah, so in this case, this is because of the Laplace mechanism. So this is because of the specific mechanism, the L1 norm it turns out to be the appropriate thing here. But there are other mechanisms for doing differential private data release that lead to other notions of sensitivity. But this is a more fundamental problem because the function that you're releasing tends to be high dimensional. Yeah, so that's a great question, which is some of the at least basic mechanisms of differential privacy don't seem to be working. So are there other notions of differential privacy, other mechanisms that would be appropriate for the GWAS setting? And I think that's an open question. So the other thing is GWASs actually have population level background information, right? So let's say I gave you the betas of an individual from a GWAS that already gives you some information about the phenotype, right? Even if the individual never participated in the data set. Differential privacy is asking how can you learn something about an individual who participated in the data set. But even before an individual participated, you can actually predict their phenotype. So there's prior knowledge. And so differential privacy doesn't actually address that. And so a question is, how do you actually take into account the prior knowledge that you're getting from population level analyses? And I think, again, we don't have a good solution there. All right. Cryptographic approaches, I don't want to say too much about this. They are orthogonal to differential privacy. So the idea is you want to do some genomic analyses with a third party, but you don't want the third party to read the genome and to figure out what your phenotype or genotype looks like. And so the idea is to encrypt and send it off to the third party. And the idea is you want the third party to work only on the encrypted data and to be able to produce meaningful results. So there's been some really exciting work in the crypto community looking at what's called homomorphic encryption schemes, which allow you to do operations directly on the encrypted data. And there's been some work, very preliminary, applying it to the GWAS setting. But again, it's very far from being practical yet. All right. So yeah, I just want to say I think it's a very hard problem, but it's really important, and I'm hoping that some of you might get interested in this.